All right, let's dive into what is basically a scientific detective story. It all starts with a really mysterious brain disease, but it ends with a discovery so out there, it challenged the very definition of life itself. This is a story of an infectious agent that completely shattered all the rules.
So, when you think about any kind of pathogen, right, a virus, a bacteria, you think of something that has genes, DNA or RNA. That's just biology 101. But what if? What if you had an infection that had no genes at all? No DNA, no RNA, nothing. I mean, according to everything we thought we knew, that should be impossible. And yet, that's exactly the baffling mystery scientists found themselves up against.
And the whole story we're unpacking today, it really all comes from this one single revolutionary paper. It was published in the journal Science way back in 1982. This is where Dr. Stanley Prusiner first laid out all the evidence for something brand new to science, something he gave a new name, but, well, we'll get to that in a minute. First, let's set the scene. Before the discovery, there was the mystery. For decades, scientists were just completely stumped by this group of really terrifying and always fatal brain diseases. They were rare, thank goodness, but they were a death sentence, and they all did the same horrifying thing to the victim's brain. There were three main culprits on the list. You had Scrapie, which affects sheep and goats. Farmers had known about this one for centuries. Then there was a very strange disease called Kuru, which was found in a small population in New Guinea and eventually linked to their funeral rituals. And finally, Creutzfeldt-Jakob disease, or CJD, a rare but devastating human brain disorder. The terrifying link? They all caused the brain to literally fill with holes, turning it into something that looked like a sponge. And here's the crucial break in the case. Scientists proved these diseases were infectious. They did these incredible experiments where they could take brain tissue from a person who died of CJD, inject it into a goat, and that goat would develop a disease that looked exactly like Scrapie. So this confirmed it. There was some kind of transmissible agent. But for the life of them, nobody could find it. It was like they were hunting a ghost. And this is where the story goes from strange to just unreal. When they tried to kill this mysterious agent using all the standard methods, it just wouldn't die. It was, for all intents and purposes, indestructible.
I mean, they threw the whole playbook at this thing. Nucleases, which are enzymes that are literally designed to chew up DNA and RNA, the agent was resistant. What about blasting it with high-powered UV radiation to scramble any genetic code? It was still resistant. They tried a whole battery of chemical cocktails, all designed to attack and destroy nucleic acids, and every single one failed.
And to put that UV resistance into perspective, to kill a typical virus, you might hit it with, a couple hundred joules of energy per square meter. To finally inactivate this Scrapie agent, it took a mind-boggling 42,000. That is just off the charts. It was incredibly resistant to the very thing that is supposed to be a silver bullet for killing pathogens by destroying their genes. So you can see the paradox here, right? This was a huge problem. All life as we know it, from the simplest little virus all the way up to you and me, it depends on nucleic acids, DNA or RNA. That's how you replicate, how you pass on information. But here was this infectious agent that could just shrug off every single attack aimed right at its genetic core. The only conclusion left, as crazy as it sounded, was maybe it didn't have one. But the agent did have a weakness. It had an Achilles heel, and finding it was the breakthrough that finally cracked the whole case wide open. If you can't kill it by attacking its genes, what else is there to attack? Well, Prusiner's team found 6 different ways to finally kill this thing. And when they looked at all of them, they realized they all had one thing in common. They were all methods known to destroy proteins. We're talking about things like special enzymes that digest proteins, or harsh chemicals like phenol and urea that cause proteins to unfold and lose their shape. The difference was just crystal clear. When they hit the agent with treatments that don't target proteins, nothing happened. It was totally stable. But the moment you use something that does target proteins, the agent became labile. Basically, it was destroyed. The evidence was overwhelming. Whatever this thing was, protein was absolutely essential for it to be infectious. So faced with this evidence, an infectious agent that was immune to attacks on genes but vulnerable to attacks on proteins, Prusiner made a really bold proposal. He said, this isn't a virus, it's not a bacterium, it's something completely new, and it needs a completely new name. He called it a prion. And the name itself is actually a clever little mashup of the words proteinaceous and infectious. It's a simple name for a really, really strange idea. an infectious protein. And this quote right here, this is taken straight from the paper's summary. It's pretty much the official birth announcement for the prion. It perfectly captures its two most important features. It's a protein that causes infection, and it's resistant to everything that's supposed to kill an infectious agent by destroying its genes. So let's just break that down to make it super clear. A virus has a protein coat, yeah, but inside it's got its genetic blueprint, DNA or RNA. A prion, as Prusiner was proposing, seems to be only protein. No nucleic acid has ever been found. This was a fundamental, paradigm-shifting change in how we understand infectious disease. Of course, a discovery this big immediately raises an even bigger, more profound question. If a prion is just a protein, and it has no genetic blueprint to copy, how on earth does it replicate? How does it make more of itself to spread the infection? In his paper, Prusiner laid out two possibilities, and both were pretty radical for 1982. The first model was, okay, maybe there is a tiny piece of genetic material hidden deep inside, protected so well that we just can't find it with our current tech. But the second model was pure heresy. It suggested that the protein replicates all by itself, with no genes at all. Maybe by finding normal, healthy proteins that are already in the host's body and somehow corrupting them, forcing them to misfold and turn into more deadly prions. And the paper ends by looking toward the future, asking a question that researchers are still trying to answer to this day. Could this exact same mechanism of misfolding proteins be the culprit behind other, much more common neurodegenerative diseases, things like Alzheimer's or Parkinson's? The discovery of the prion didn't just solve one medical mystery. It blew the doors wide open on a whole new field of science, forcing us to completely rethink the fundamental rules of infection.